fundamentals of enzymes and catalysis, let's talk about how we can classify enzymes based on the type of reaction that they catalyze and recognize those types of reactions when we see them. We'll do some examples, go over some concepts, all that good stuff. Now, different enzymes can catalyze different reactions in the same or different ways. So remember, we have a lot of different enzymes, but at their heart, they all kind of like share similar basic strategies that we can talk about. And because we have so many different enzymes, in part so that we can have specificity, so that we can have all those different enzymes that can only recognize and work on certain things, so that you can then regulate what's happening when. You can also have different enzymes catalyze the same reaction in different ways. So remember, the enzymes are kind of like changing the path that things are taking, but they're not changing the input or the output. I want you to be able to look at substrate and products with or without a reaction mechanism actually drawn out, and be able to tell what type of change is occurring. So you have to kind of look at the before and after. It's one of those, it's kind of like one of those pictures, like spot the difference between these pictures and like circle the differences in your, in the newspaper, like cartoon section or whatever. It's like that. We need to figure out what's changing between our substrates and between our products. And these are the main types of changes that I want you to be able to identify. They're roughly classified by their enzyme commission classification, and so I'll talk more about this in a minute. But these basic types are ligation. So we'll talk, this is like joining things together with like the help of ATP. Hydrolysis, so we're going to be breaking things with the help of water. Transferring groups, isomerization, so switching around the order of things, redox, and elimination and bond breaking in other ways. So we'll talk about like lyases. So as I mentioned, things are kind of like grouped by their enzyme commission numbers. So the enzyme commission numbers, this is kind of the way we give each enzyme a unique ID that then we can go and look up and find information about. So if you're interested in these, I can share more information. I have a whole video about these and things like why we need it, because people were naming things old yellow enzyme and new yellow enzyme. So we needed some order and some classification. And so we have these 7 main classes, and the enzyme commission number is going to be like this 4-part number, where the first number is the main class. Okay, so just a quick class and enzyme classes. So I don't necessarily need you to know like all of these different names, but I do want you to recognize what type of reactions are happening when you look at the substrates and the products, or when you look at a reaction mechanism drawn out. So these are going to be oxidoreductases. These are going to catalyze our redox reactions. We'll have transferases, so these are going to transfer functional groups such as amino or methyl groups or phosphoryl groups. Hydrolases, these are going to use water to break bonds. Lyases, these are going to add or remove things. These are kind of trickier. They're going to add or remove things to make or break bonds. You'll often see them doing this with double bonds, but not always. But the key is that they're going to do it by mechanisms other than like using water or using redox. Isomerases, these are going to help molecules rearrange their atoms. Remember, like when we talk about like cysteine or proline can go with cis and trans. Well, that's helped out with the cis proline isomerase. Ligases, these are going to join 2 molecules together with the help of a nucleotide triphosphate, such as ATP. And translocases, these are like a super new class, so you might not even see them listed in your textbook, but these basically move things like ions, often across a membrane, but they're doing something active. It's not just like a pore. You can often get hints to what type of reaction enzyme catalyzes by looking at its name. So remember oxidation and reduction. Oxidation is loss of electrons. Reduction is gain of electrons. And so if you see the word oxidase, you see the word dehydrogenase, oxygenase, peroxidases, these are basically going to be oxidoreductases. And so that's one hint that oxidation or reduction is occurring. And we'll look at some more examples, especially when we get into metabolism, how typically if you see something is being oxidized, you're going to see a gain of bonds to oxygen or loss of bonds to hydrogen. And if something is being reduced, you'll see a loss of bonds to oxygen and or a gain of bonds to hydrogen. And so you'll see enzymes that are going to be named these sorts of things. Transferases, here we're like transferring functional groups. We could be transferring a methyl. We could be transferring an amino group, like the amino group. We could be transferring like our kinases. That was transferring that phosphoryl group from ATP to a substrate. And then we can have things like phosphorylases, which are more complicated because they transfer phosphate from inorganic phosphate. They transfer the phosphoryl group from inorganic phosphate to a substrate. But those would all be examples of transferases. They're transferring kind of like big groups from one place to another, from one molecule to another. We also have hydrolases. So basically, they're going to use water to help cleave bonds. And so phosphatases are going to cleave off those phosphate groups. Phosphodiesterases are going to like cleave those DNA letters apart. Proteases, these are going to cleave our proteins apart. All of those would be examples of hydrolases. They're turning water into molecular scissors. Lyases, basically what these are going to do is those are those weird things that change bonds without, they make or break bonds without using water, without using redox. We'll see some examples like decarboxylases, aldolases, synthases. And so we'll see things like we're often going to be characterized by like elimination reactions, because these elimination reactions, well, you think about elimination, forming double bonds, lyase. Isomerases, these are either going to like change between stereoisomers, which would be like a racemase, or what typically we're seeing mutases, which are going to shift groups around within a molecule, changing what's linking where, but not actually changing the this like RNS stereoisomers. Which is good because that means that we can tell apart on a figure if we kind of look and see where which bonds are connected to what. If the number of atoms isn't changing, but the location of, say, a double bond is shifting from one place to another, that would be an example of an isomerization. And a ligase, well, here we're kind of bringing things together with the help of ATP. You might see carboxylases, you might see synthetases, and we'll look at some examples of this. Sometimes they're just named a ligase, which is nice, like DNA ligase. Okay, and then translocases, don't worry about these. Just like know that it's this new class, and they like to move it. Okay, so now let's try to classify some reactions. We're going to classify the different steps of glycolysis. So we'll talk more about glycolysis, much more about glycolysis when we get into metabolism, but it's going to be the process of breaking down glucose or blood sugar. And so we're going to try to identify what types of reactions are occurring just by looking at the different products in the pathway. And note that when we talk about metabolism, we're going to be talking about like intermediates. So we can think about like each of these molecules as an intermediate in the overall glycolysis pathway. But these are not like reaction intermediates. So you would have like a reaction coordinate diagram for each of these reactions that is happening. Or you could put them all together though, remember, because what's happening is to get from the glucose to the pyruvate, the difference in the free energy from here to here is going to be the same no matter what pathway you take in between them. Because the enzyme cannot change that overall delta G, but it can make it easier to get there. And also by doing it in all these steps, now you have all of these different products that you can siphon off into other pathways or use for things like that. So we'll see metabolism's really, really cool. It can get really, really complicated, but we can just kind of look at the molecules to see what type of reaction might be happening. Okay, so let's go ahead and do that. Let's kind of try to figure out, okay, are we looking at a group transfer? Are we looking at isomerization? Are we looking at a redox reaction? Are we looking at elimination? What's going on in each step? So let's start with number one. Here we're going from glucose to glucose 6-phosphate. And it's telling us that we're basically also going to take ATP and produce ADP and an inorganic phosphate. So what type of reaction is taking place? Right, that's going to be a group transfer. We're transferring that phosphoryl group from ATP onto glucose. And so this is taking place with a kinase, and this is the name of this kinase is hexokinase. So when you're looking at a one of these like reaction arrows for an enzyme, basically what you'll see is that underneath the arrow is, or on top of the arrow, by the arrow, you'll see the enzyme name. And then you'll also like see any sort of cofactors that are needed. So we're going from glucose to glucose 6-phosphate. Now what's going to happen between our glucose 6-phosphate and our fructose 6-phosphate? It's kind of hard to see on the little figure, but what's happening is that you're kind of shifting the position of this double bond. So if we here you had a double bond to oxygen here, you had a carbonyl here, and now you have your carbonyl here. So what type of reaction is happening? Yeah, that's going to be an isomerization. And so the reason why I was hiding the enzyme names is because sometimes they give it away. So this would be a phosphoglucose isomerase that's going to be catalyzing this interconversion. Okay, now what's happening between fructose 6-phosphate and fructose 1,6-bisphosphate? And note that we use this term like bis when we're talking about like if something has like bisphosphate, that would have 2 phosphate groups, but that aren't attached to one another. If you talk about like diphosphate, like a nucleotide diphosphate, that would be 2 phosphate groups that were attached to one another. But when you see bis, that's basically they're saying they're in different places. And so here we had 1 phosphate group. Here we had 2 phosphate groups. What type of reaction just happened? Yeah, we just had a group transfer. We had a functional group transfer of that phosphoryl group from ATP onto our fructose 6-phosphate. Now what we're going to do, we're going to take this fructose 1,6-bisphosphate and we're going to convert it into 2 molecules of dihydroxyacetone phosphate. So this one's more complicated. So we can see that we're breaking things, but we're not going to be using water. We're not going to be using redox. Instead, basically what we're going to be doing is this is going to be a lyase. Don't worry about it, but it's called fructose bisphosphase aldolase, and we'll talk more about it later in the semester. Okay, now you can do this one though. We're going from dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. What type of reaction is happening here? We're just shifting things around. We're just moving the orientation of things. We're not changing the number of atoms, just their position. This is an example of an isomerase. So this is going to be triose phosphate isomerase. Okay, what about now? We're going from glyceraldehyde 3-phosphate to 1,3-bisphosphoglyceride. What are we doing? Here, this is a trickier one. So basically we are adding a phosphate group, but we can see that we're also going to be reducing NAD+. And so this would be an example of both an oxidation and a group transfer. And so you can see that it's happening via glyceraldehyde 3-phosphate dehydrogenase. So remember, dehydrogenase is one of those symbols that we have some sort of redox reaction happening. Okay, so now we go from 1,3-bisphosphoglycerate to 3-phosphoglycerate. So remember, bisphospho, we're saying we have 2 phosphate groups, and now we just have the 1. So we've lost a phosphate group. What type of reaction just happens? We just had a group transfer. We're transferring the phosphate group from 1,3-bisphosphoglycerate onto ADP to get 3-phosphoglycerate. So this is done by phosphoglycerate kinase. And so basically, this is named in terms of the reverse direction, because remember that enzymes, they do things in both directions. They can't change that. Sometimes we might show a one-way arrow, but that's just because something is so favorable in one direction, or the concentrations are so out of whack that it's practically irreversible. But the enzyme itself can catalyze things in both directions, and so we can talk about this as being a phosphoglycerate kinase in terms of thinking about going from 3-phosphoglycerate to 1,3-bisphosphoglycerate. Okay, so what's happening in this one? Now we're going to go from 3-phosphoglycerate to 2-phosphoglycerate. What's changing here? Yeah, we're moving the position of the phosphate group. And what do we call it when we move the position of the phosphate of the group? that's just going to be an isomerization. So this is going to be done by phosphoglycerate mutase. So remember, mutase, that's one of our words that hints at us that we have a isomerization reaction happening. Okay, now let's go from 2-phosphoglycerate to 2-phosphoenol pyruvate. What's happening here? Well, we can see that we're forming a double bond, and we're kicking out water. So we have an elimination reaction, and this is going to be done by some sort of lyase. So we have an elimination done by enolase. Don't worry too much about the lyase things, just know that we have this elimination happening. Elimination of water, we're forming a double bond. And now finally, how are we going to go to from phosphoenol pyruvate to pyruvate? Well, it's telling us that we can see we're losing a phosphate group. It's telling us that we're transferring that phosphate group to ADP to make ATP. And so what type of reaction is this going to be happening? This is going to be, right, that's going to be a group transfer, and it's carried out by this kinase, pyruvate kinase. Okay, so let's look at a couple of examples involving proteases or peptidases. And so basically we'll look in more detail at like serine proteases, but it's going to be similar to how cysteine and threonine proteases do things. And the naming here is talking about the enzyme, the catalytic residue. So which enzyme in the active site is actually going to be doing the work, and then they're not where it's actually going to be cut. We also have like aspartic proteases, glutamic proteases, and metalloproteases. And these guys are going to they're not going to go through covalent intermediates. Instead, they're just going to activate water. So let's look in a little more detail about our serine proteases. Basically, it has this cool catalytic triad. So triad, there's 3 catalytic residues. We've got a serine, a histidine, and an aspartate. And what's going to happen is we're going to get this oxygen to act as a nucleophile and attack the carbonyl carbon. But the serine, remember, isn't going to be a very good nucleophile as is. Because remember that the serine is not normally going to be deprotonated because it's got this high pKa of about 13. So we need to actually lower that pKa. So that would be the pKa in isolation, but in the context of the active site, we can actually lower this. And so how do we lower the pKa? Well, we lower the pKa by helping stabilize the negative charge, helping pull away the proton. These things would lower our pKa. And so we're going to find that we're going to have serine get its proton stolen by histidine, and histidine is going to help make more basic, more wanting to take that proton by an aspartate being nearby. So what you have is basically you have the pocket on the enzyme in the active site where there's like a substrate binding pot. And this is binding there is going to get that trio into position to get to work. So you wouldn't want things always to be activated, always on all the time. Instead, you only want to activate it when you have a substrate bind. So when the right substrate binds, it's going to fit nicely in the pocket of that enzyme. And the enzyme, remember, what's it going to stabilize? It wants to stabilize the transition state. And so basically, you're going to have that induced fit. The induced fit is going to help make it so that that trio is right in position to get to work. So the first things first, we got to pull off that proton from serine. And so how are we going to do this? We're going to do this using a histidine. But histidine, well, that's not a very good base, remember? And so how are we going to get histidine to be a good enough base to pull off a proton from something that's not a very good acid? This is where the aspartate comes in, because remember, aspartic acid is a great acid. It really wants to be in this conjugate base form. It really wants to be in this negative form. In this form, it can hydrogen bond with with one of the amines in the histidine. By forming this hydrogen bond, what's going to happen is that when the histidine gets protonated, it's better able to stabilize that charge. So it has this kind of negative charge that can help make it stabilize this protonated form. And so the histidine is then going to be able to take that proton. Once you've taken the proton, well, now you've got a strong negative. You've got a strong nucleophile. That serine really doesn't want to be negative, so it's going to attack that carbonyl carbon. Now remember that the enzyme is helping everything be held in the right orientation to do this. It'll attack the carbonyl carbon, We get our tetrahedral intermediates. When we attack a carbonyl carbon, we get a tetrahedral intermediate. It is a real intermediate. It's not one of those transition states, but we did have a transition state in order to get here. So we had to form a bond to the carbon, and then we had to break this bond, this double bond, so to get to this tetrahedral intermediate. So in between those, we would have had a transition state where we would have those partial bond formations. But what we have is basically we have this tetrahedral intermediate. And it's attached to our substrate in this case is covalently attached to the enzyme because the serine is coming from the enzyme. This is our substrate. But we haven't broken our substrate yet. We've only broken one of those double bonds. And so now we need to break this off. And we want to make sure that we break it off the right way too. The enzyme is going to help in a couple different ways. Basically, there's this part on the enzyme, this called this like oxyanion hole. So oxyanion, because it has this negatively charged oxygen. And then there's going to be the amide nitrogens in the backbone of the peptide in this pocket are going to help stabilize this intermediate and kind of make sure that it's going to break the right way. So this intermediate is going to break down. What's going to happen is that the nitrogen in what was the peptide bond is going to steal a proton from that histidine. And so this is going to be stealing this proton. This is going to break the first part off, but the N-terminal part is still stuck on the other part. So you still have part that's stuck to the enzyme, and then you have part that has been broken off. So we need to, in order to use this enzyme again, we need to regenerate it. We need to basically kick off this other part so that we can get this, to do things over again. What are we going to call this? Right, this is going to be one of those general acid-base reactions. And in this case, we're going to be having a general base because this nitrogen is going to be stealing a proton, making this hydroxyl ion that can then go and attack the carbonyl carbon. You get the whole situation happening again, but this time, because you're doing it from water and not doing it from the amino acid, you're going to be breaking things off. And so you're releasing the rest of your product. And if you were to go and look at one of those reaction coordinate diagrams, you would see that you do have all of those intermediates formed, and you do have all those transition states, and you get this like weird looking graph like this. And you can see that we have kind of like two transition states that are pretty similar because we have to do that like reaction state twice. So what types of catalysis was that an example of? There were a couple of different types we could say. Yeah, so it was a covalent catalysis, and it was also going to be a general base catalysis. Some of the proteases don't go through those covalent intermediates. Instead, they just use water both times. And so an example of this is our aspartic proteases, where basically you have these two aspartates, and one of them is going to steal a proton. It acts as a general base, it steals a proton, makes a hydroxyl ion that can then go and attack. And then you do the same thing again using your other aspartate in the pocket and regenerate things, using that water to kind of help break things apart. So some other proteases just like skip the covalent intermediate and instead use water both times. So an example of this is an aspartic protease. And so they're basically going to have an aspartate act as a general base. So remember, it can get confusing because it's acting as a base, but we call it an acidic amino acid. And we call it an acidic amino acid because it's so good at acting as an acid. Its neutral state acts as an acid. And then we get this we get it normally in our conjugate base form, which isn't a very good strong base, but in the context of this active site pocket, when you have this other aspartate right next door, with another negative charge, it's kind of like making that positive charge look a little more appealing. You can get it take a proton, and then you have a hydroxyl ion that's going to go attack the carbonyl carbon, and then you basically have things get rearranged from that tetrahedral intermediate. Take that proton back, and then basically break your peptide in two. And in this case, you didn't have any covalent intermediate. You were just able to release the pieces. And so here's an example of like how you might see this in a paper. You get some reaction coordinate diagram like this. This is showing you a couple of different aspartic proteases. And so here's the active site of pepsin, one of the proteases. You can see the two aspartates here. Here's the two aspartates in HIV protease. So these are real enzymes doing real stuff, and you can actually see that this is where the catalysis is happening. So we have our active site where the substrate is binding and the action's happening. And then we can talk about catalytic residues. So these two aspartates that are actually directly participating in things. But then you can think about all of the different weak interactions that are working to hold this substrate in place. And what's going to be our rate limiting step? Right, we want to look at the delta G double dagger of the biggest step, the biggest one. And this will be to get to our transition state 1, to get to that point at which we're actually doing that first attacking. So what type of catalysis was this an example of? Right, that was another example of our general base catalysis, where basically we're stealing a proton from the water, getting it to attack, and then this tetrahedral intermediate collapses. And the amino acid leaving group is protonated as it's expelled. And when it gets protonated, basically now you're resetting the system to where it was. Because remember, enzymes can do the same thing over and over and over again. That's why we call them catalysts. So I know that was a lot of information. We're going to be doing a lot of examples in class. Again, you don't need to worry about the specific mechanisms and being able to draw out the specific mechanisms of every enzyme, but rather I want you to be able to kind of follow the flow in these diagrams and be able to kind of identify the changes that are going on and some of the strategies that enzymes are using to help make those changes possible.